Thanks to the organisers for giving me the chance to present. I'm going to talk to you about finding regulatory variants. So, as we all know, many important phenotypes and diseases are driven by differences in gene expression rather than by differences in protein sequence. But it can be very challenging to identify exactly which variants are involved. Statistical methods, statistical association methods, such as GWAS, are very powerful and can link phenotype to a particular region of the genome, but they can rarely pin down exactly the causal variant because there's so much genetic linkage between variants. And these methods mainly target a single nucleotide variation. And when we think about other types of variants, such as insertion-deletion mutations and longer and more complex structural variants, these involve more bases and may often have a larger impact on gene expression than SNPs, but can be even harder to find and test where they have an effect. One reason for this is that it can be challenging to map short reads that contain these variants onto a reference genome. So this gives you problems identifying them and then for genotyping them in further samples. And also, GWAS tends to work best with variants that are quite common in the population, whereas these longer variants tend to be quite rare. Massively parallel reporter assay tried to deal with some of these issues by looking directly at the effects of DNA sequences on transcription. So the type of MPRA that we've been using is called the Survey of Regulatory Elements, or SURE, and is described in these papers here. This approach works by taking your entire genome of interest, breaking it into fragments, and inserting fragments of a suitable size into a plasmid, each of which has got a unique barcode and a reporter gene, but does not have a promoter. So this gives you a library of plasmids where you'll only get transcription of the reporter gene if your fragment from the genome has got an active transcription start site. So these are transfected into a suitable cell line, and then plasmids that have got an active fragment can fire off transcription. So we can see that plasmids that have got the purple barcode are giving you lots of transcription, and the ones with the brown barcode, not very much. So to quantify this, we sequence transcripts from the transfection and also the input library. So just to look in a bit more detail at the quantification, this is the part of the plasmid that's sequenced. So one set of data, the input PCR, so sequencing the plasmid library. This gives you paired-end sequencing data, which contains both the sequence of the genomic DNA insert and the barcode. And you then align this to the reference genome to find out where the genomic DNA fragment came from, and also if it carries, if it covers variant sequence, which allele it carries. And also we count how many times each of these barcode plasmids is sequenced in the library, because if you have a sequence appearing more times in the input library, you'll expect it more times in the output, and we want to correct for this. So the second type of sequencing is once you have transfected your plasmid library into cells, to sequence the transcripts that are produced to find out what barcodes they carry. And for this quantification, we just count how many times each barcode is sequenced, and this gives you a measure of the transcription activity of the fragment that it carries. So we then use the barcode to say that a genomic DNA fragment from this part of the genome carries, has this much activity in the assay. And also we can use it to compare fragments that cover the same region if some contain allele A and allele B, and to compare the activity of those sequences. The aim of SURE in our group, we're doing it mainly in cattle to identify loci that are linked to important production and disease phenotypes in cattle, and also to study how this regulation is conserved across mammals. There are two subspecies of cattle arising from independent domestication events, and we've produced a SURE library from one animal from each of these subspecies, so one bus taurus and one bus indicus, and also a SURE library from a human sample for comparative analyses. These libraries were transfected together into primary aortic endothelial cells from cattle, and also into the same cell type from humans to see if there are any variants that have different effects depending on the context. My project focuses on adapting the SURE pipeline, which is mainly developed to study SNPs, to study indels and larger structural variants. And one large change that I have made to try and improve coverage of longer variants is to use a graph genome instead of a linear reference genome to find fragments. you're not familiar with graph genomes, they consist of nodes that represent DNA sequence and edges showing how they're connected together in each genome. In this very small toy example, we have two genomes, blue and orange. They both pass through this node here, so they both contain this sequence. Then we have a 2 base pair substitution, and then over here we have a node, sorry, not speaking to the microphone, we have a node that blue passes through and orange just skips across, so this represents an insertion or a deletion. So one advantage of a graph genome in this context is that reads that have got a large difference from the reference have got a better chance of being aligned correctly. So in our example, if we used a linear reference from the blue genome, just looked at that, then a read from the orange genome might not be aligned correctly. Whereas if we have the graph genome, then they're both there and it can be put in the correct position. So we have generated haplotype resolved genome assemblies for each of the 2 animals that went into the SURE libraries and aligned these with the reference genome for cattle to make a cattle graph genome. And done something similar for the human SURE sample and the human reference, except using a published haplotype resolved genome for the human sample. So here is my SURE pipeline. The fragment locations are identified by aligning the input PCR reads to the graph genome, and then these are combined with the cDNA counts as before. Then for each variant that was identified in the graph genome, I found fragments corresponding to each allele, and then use a regression model to test whether which allele is carried by the fragment affects how much transcription activity it's got in the SURE assay. And I'm now going to talk through some results from analysing the 2 cattle libraries using this pipeline. So here I'm showing results from the cattle libraries transfected into cattle cells. And this is for all indels of any size that we were able to test. In this plot, each bubble represents one variant, and the size of the bubble tells you how many bases were inserted or deleted. On the y-axis, I've just got the p-value, and on the x-axis is the ratio of expression activity between the 2 alleles. So one's on this side that have got a positive value. This means that fragments that carry the reference allele have a higher mean activity in the SURE assay than fragments carrying the alternative allele. And on the other side of 0, it's the opposite, so the alternative has more activity. So using this pipeline, we're able to look at a lot of this kind of variance and also have pulled out quite a few that are significant that we can look into further. I've started looking into what are the important features about these variants. So here I'm looking at the variant size and seeing that larger variants, so inserting or deleting more bases, seem to be more likely to have an effect in the SURE assay. But this is just in early stages. I'm now going to show you how we can zoom in on an individual variant. So this is a medium-sized deletion with a very significant p-value. The 2 boxes here are 2 replicates of the transfection into cattle cells. And for these, the fragments have been binned according to their activity in the SURE assay, with fragments that have got the reference allele in the top plot and ones that have got the alternative in the bottom plot. And this bottom plot has got an inverted y-axis, so high expression is down here. On the x-axis is progression through the graph, with nodes shown in alternating shades of grey. And then for the intensity of colour tells you how many fragments pass through a particular node and have a particular level of activity. So this node here represents the sequence that is deleted in the alternative allele. And you can see indeed that none of the alt fragments pass through here. So from these plots, we can see that in both transfections, fragments with the alternative allele tend to have higher transcription activity. So these plots let us look at some of the details of the variant and also see how consistent they are between transfections. And here are the results, again for indels, for transfecting the same cattle libraries now into human cells. Unfortunately, these transfections didn't work as well, so we didn't capture as much total transcription. So the number of variants that cross this threshold for significance is lower. However, there are enough variants with plenty of data that we're able to compare the activity of fragments carrying the variant, to compare their activity in each cell species. What I'm showing here is for each variant, the estimated effect by the model of what it has in human cells, and then subtracting its estimated effect in cattle cells. So you can see that there certainly are some differences. And then on the y-axis is a p-value that's calculated using the estimates and standard errors from the model. I'll just finish off by showing some results for more complex variants. So these have more than one alternative allele, and these also tend to have more complex paths through the graph. So these don't work with my standard model because that assumes just 2 variants. So what I've done instead is to fit 2 models, one that includes allele identity as a covariate and one that doesn't, and then just test whether including the allele in the model gives you a better fit. In this density plot, I show the ratio between the whichever allele has got the highest expression over whichever has the lowest. And to show you an example of what we're looking at, this is the tiny part of the genome graph that represents the variant. First of all, looking at the reference, it skips straight from this node to this node, and fragments that carry this sequence tend to have pretty low activity in the Sure assay. However, when we look at the alternative allele, it's got a 19 base and a 1 base pair insertion relative to the reference, and these fragments have much higher activity. And then we have the second alternative allele with the same 19 base insertion and a different 1 base insertion, and these ones also have much higher activity. So this is something that we're able to look at with our approach that can be quite difficult to study using other methods. So as I said, I've told you just about the results for transfecting the cattle lab. But now that I've built the pipeline, we'll go back and do the same with human genomic DNA libraries and see what's conserved and what isn't. Also in the group, we have a set of ProCap data, which identifies active transcription start sites by sequencing nascent transcripts. And this is coming from cultured cells, so they're actually growing in their sort of biological context. And this complements a weakness of MPRA, which is that it takes the sequences out of their biological context. But we're going to be able to put the two together and see which variants that are significant in Sure are actually near to ProCap peaks. So tell which ones are most likely to be biologically relevant. And finally, with the genome results, I'll be looking at what influences whether a variant is likely to have an effect. So I've talked about variant size. We can also look at variant frequency, variant type, how far it is from a transcription start site, and various other effects. So finally, I'll say thank you to everyone that's been involved in this project at Roslin, especially James Prendergast, who leads the group, and Rachel Owen, who carried out most of the lab work. Also to these 2 authors of the original paper, who now do the assay commercially and perform several of the steps for us. And of course to our funders. And thanks to you for your attention.
Thank you. We have time for questions from the audience.
Yeah, thank you. Very interesting method and talk. How do you select the variants you're looking at from all of the ones that are available, basically?
So we are trying to look at all of the ones that are available. So there are, I think we found about 1.8 million indels between these 2 cattle subspecies. So we've tried to test all of them. Some of them we didn't get enough. We didn't get fragments for, we didn't get enough transcription for, but we're not doing any selection until we're looking at the analysis. Yeah. We've also done it for SNPs, which is a different project, and they're studying sort of 16, 17 million in the same assay. But yes, once you process the results, it's all sort of the same data.
Other questions? Actually, I have a question. So when you do the model that evaluates the different type of complex type of variants, Some of them can be obviously a problem of the alignment. Is there any way you can actually have a feedback about the possible? So I mean, I don't know, the method I expect that returns some sort of probability, right? I mean, have you tried to test actually if there are some mistakes or something that is due to the bad alignment?
Not directly, but we have sort of a number of filters in the pipeline that will tend to remove ones that are not real. So for example, when we are counting up the fragments from each allele, if the number of fragments you get don't approximately match what you're expecting from the genotypes, then we'll discard that. So for example, for an animal that's a heterozygote, you should get about 50-50. Or if it's a homozygote, you should get all from one allele. And if you are getting something that diverges from that by very much, then as you're saying, it might well be due to a problem finding the variant. So we've got things like that that should discard these, but we haven't got anything like directly testing.
Okay. Other question? Okay, if not, thank you again, Lindsey. And we can move on. Let's talk.